Dolphins are top predators in marine ecosystem, and therefore understanding their foraging ecology can be highly informative, not only by learning their ontogenetic dietary shifts, but actually also by unraveling their trophic relationships. Traditionally, the way to learn about dolphin foraging was through analysis of the stomach content of dead individuals that were washed ashore. This early approach provided early snapshots of their dietary intake. Nowadays, we know that annual deposits of growth layer groups in dolphin teeth reflect their diet at the time the deposit was formed. And that means that dolphin tooth can actually function as a temporal archive of their diet. Thanks to the recent advances in the application of stable isotope analysis, we can now use quantifiable measures of long-term dietary intake to investigate relationships between these animals and their ecosystem. And as dolphins are among the top marine predators, this way we can actually use them as bioindicators of the region's environmental history. For places such as Hong Kong and the Pearl River estuary, which have been heavily urbanized in the past several decades, this can be an important tool to study environmental change.
Part of my postgraduate research focuses on stable isotope analysis on Chinese white dolphin teeth. It might come as a surprise to see how much biological information is archived within a dolphin tooth. It is almost as reading an ecological history of their life. The dentine within a tooth, which is deposited year by year, tells how old the animal is and indicated its nutritional history. This deposition are synthesis of bile appetite, which deposit within the tooth cavity until it's fully filled up. I use a fine micro-drilling apparatus to mill each growth layer groups of dentine samples and test them with stable isotope analysis. This approach can tell us a lot about the ontogenetic dietary shifts through an individual's life, in which the delta-15 nitrogen changes as the individuals consume higher trophic level diet. To put it simple, if I am a vegetarian, I would have consumed lower trophic level diet, resulting in lower delta-15 nitrogen. But if I consume more meat, then the delta-15 nitrogen will increase. That's how I investigate the dietary shift throughout the life of Chinese white dolphin through just the small teeth. I can also find out the weaning age of the dolphin calves. In a similar fashion, we can also investigate the environmental change in the peripheral estuary, analyzing nitrogenous pollution. The crown tip isotope is formed prior to 1 year of age and are influenced exclusively by the foraging ecology of the mother. Therefore, this can reflect how the environment changes in response to the influx and anthropogenic overload of nutrients, much of it due to the increasing population.